Welcome biologists to this screencast that will go over genetic drift and gene flow, which is from chapter 9C of your textbook. So our learning intention is to learn how change by chance can alter genetic makeup of populations. And by the end, our success criteria will be defined by being able to describe the bottleneck effect, the founder effect, and what we mean by gene flow, which is in effect migration, and how that can lead to changes in allele frequency. You'll also be able to discuss what the effects of these changes can have on the gene pool overall. Your study design, the two key dot points we're looking at here. What are the causes of changing allele frequencies in a gene pool, including environmental selection, which we've already covered, genetic drift, and gene flow, which are the ones that we're talking about here. And again, we've already talked about mutations. So the main focus here is this genetic drift and gene flow. And you need to be able to describe the biological consequences of changing allele frequencies. So we are talking about change by chance. These are purely chance events. There's no selection involved. So this is not natural selection where you're talking about selection pressures. However, the end result is that the gene pool is still altered. It's unpredictable and it can vary from generation to generation. And if your starting population is smaller, it's more likely to have a greater impact. This video sums up the key things we need to know about in terms of bottleneck and founder effects and gene flow. Only goes for a couple of minutes, so I'll play this, which will help describe what we're about to discuss. Then I'll go through each of them step by step with you. And as always, when we meet, if there's any questions, I hope that we can go through those. But I'll play this video for you now, which should hopefully cover a fair amount of the material that we need to know.
Thanks for stopping by. Today we're going to talk about genetic drift. So genetic drift is a mechanism of evolution that's responsible for random changes in a gene pool. All right, it happens to all populations, but it has a much more visible and drastic effect in small populations. Right? It's driven by chance and not selection, so evolutionary fitness doesn't matter here. It's all random. Natural selection doesn't come into play when we're looking at genetic drift. All right, and there's two main types of genetic drift. You have the founder effect and the bottleneck effect, and we'll look at some examples of both of these. So let's say that you have a town, call it Darwinville, and you have 100 people that live in this town. And of those 100 people, 95 of them are right-handed, and the other 5 are left-handed. And let's say a small group of about 4 people from this Darwinville decide to move away and settle some new uninhabited land. And let's say these 4 individuals happen to all be left-handed. All right, their gene pool is now drastically different from the group they left because all their descendants will be left-handed. All right, this is an example of the founder effect, when a small group leaves from a population and carries with it a new gene frequency. Okay, so let's go back to our town of Darwinville, where we now have 96 people and only 1 lefty. All right, let's say a freak storm comes through and kills 75 of the residents of Darwinville, including our last left-handed person and all remaining recessive left-handed alleles. Right, and then the new population, now 21 individuals, will have no left-handed posterity from this point on. So this is an example of the bottleneck effect when there's a random reduction of the population due to some random event. All right, so hopefully this video and these examples were helpful. You can see the link in the description for more information and for more examples on genetic drift, or you can hit me up in the comments if you have any questions. And be sure to check out my other videos and let me know if you have a specific video that you want me to make. Thanks for watching. We'll catch you next time.
All right, so let's go through some definitions. You should add these to your Quizlet as you go. Genetic drift is a random chance event that significantly alters the gene pool. All right, so again, it's random by chance, but it leads to a change in the gene pool. So that's what we mean by genetic drift. Make sure you've got your definitions. So from your textbook, the bottleneck effect, typically that is associated with some sort of significant natural disaster. So this image taken from page 506 shows you the original population. You've got a huge variation or genetic variation there, as illustrated by multiple different colors of whatever this bug is, beetle. There we go, it's a beetle. So multiple different colors of beetle. The bottleneck is basically a metaphor for squeezing the population through some sort of natural disaster. So basically not every individual in the population can survive this natural disaster. So you have this bottleneck where some alleles squeeze through, others are wiped out. So the surviving population is no longer representative of the initial gene pool. In other words, instead of having all of this variation, only a couple of variants made it through. And so this population no longer represents the starting population. And so as that population grows and breeds, you can see the pink beetle's gone, the blue beetle's gone. You've only got these green and yellow beetles left. So there's a significant reduction in genetic variability and obviously an alteration to the gene pool from the original population. Same principle, just illustrated slightly differently. See if that helps. Again, you've got a bunch of different colors representing different phenotypes, genetically diverse. All right, you hit your bottleneck and only the blue and yellow ones survive and come out the other side. When you come out the other side, the only blue and yellows, they'll breed and that's fine, but you won't get the genetic diversity that you once had because you can't, because they died out as a result of this natural disaster. So do remember, bottleneck effects are typically associated with natural disasters. That's often indicated in questions on Saxon exams. There's some sort of natural disaster. What's the name given to this phenomenon? The correct answer will be bottleneck effect. So be aware of that. Keep that in mind. So bottleneck effect, just in summary, significant disaster reduces the population. The remainder may be unrepresentative of the original gene pool. The population will rebuild, but the alleles will be different in terms of the allele frequency to the original population. Therefore, the phenotypes represented will also be different, and diversity will almost certainly be impacted. To give an example, there's an article here which you're welcome to read if you wish. You'll have access to this PowerPoint, so you can just click on the link if you want to follow it instead of having to type that. But if you have an interest, you might want to have a bit of a read. It's from Nature magazine. But this refers to Tasmanian devils, which we know have very low genetic diversity, which is impacting with respect to the facial tumour disease. Because they don't have enough genetic variation, this disease is wiping them out. But over history, and we're talking ancient history, geological history, there's been at least 3 crashes in their population where they've almost been wiped out, resulting in genetic bottlenecks. And so these 3 crashes are bottleneck effects that have occurred since Tasmanian devils were isolated on Tasmania. In other words, once they got wiped out on the mainland and were only on the island of Tasmania, 3 times their population has been reduced through natural disasters, leading to significant reductions in genetic diversity. If you want to read more about that, the article's there. The founder effect, again, this is done by chance, but you'll have a population, again, with variation in it. One population breaks off and starts a new colony or a new population in a new location. In other words, it founds a new city, a new suburb, a new whatever, right? But it moves out and starts out on its own. Again, it's not representative of the initial population, because it's only these green beetles and all the others are left behind. And therefore, the future generations are no longer representative of the genetic diversity in the original population. So it's literally a small cross-section, moves out, and starts a new population. And just like before, here's that similar sort of scenario with a different illustration to show you the same thing. Lots of genetic diversity. This small subgroup moves out, or a founding group goes out and starts a new colony. And this population now is no longer representative of the original population. It's genetic drift, okay? The genetic diversity drifts and changes over time as a result of chance events. Bottlenecks are one, founder effects are another. So you need to know both of those. And so again, in summary, the founder effect, a few members of a larger population break off and start a new, smaller population. The smaller size reduces the amount of genetic variation, and it's likely to be non-random. So dangers of genetic diversity, you should be able to answer this. So before I present them, I encourage you to maybe stop the video or take a moment and see if you can answer that already. But you should know the answer, which I'm obviously going to reveal in the video in a second, but have a think first. But inbreeding in a small population means that if there are harmful alleles or deleterious alleles in the population, they're still going to be passed on. They're still going to survive because there's not enough individuals to breed them out. So it means that overall, the health of the population may suffer. And you'll have reduced adaptive ability. So in other words, if there's a new selection pressure comes along, like the facial tumor disease for Tasmanian devils, because you've got low genetic diversity, there's less likelihood that your species can survive and adapt to this new selection pressure. And the risk is that you get wiped out. The last bit you need to know about is gene flow. There's not really much I'm going to put in here about this. Really, gene flow is just migration. You need to know the difference between immigration and emigration. And again, I would put those into your Quizlet to help build your vocab and know those definitions. But immigration just means that a new allele moves into a population, IMI meaning in, and emigration means that it moves out. So I for in, E for exit. All right, immigration in, emigration with an E, exit. It is literally just the movement of alleles between populations, and that movement can obviously change the makeup of a gene pool. You'll get new alleles moving in or old alleles moving out. It changes the ultimate allele frequency within the gene pool and can change genetic diversity. All right, so in summary, we now should know the three main mechanisms that occur by chance that can alter genetic makeup: bottleneck effect, founder effect, and gene flow, migration. You should be able to describe all of those. You should be able to describe how they can change the allele frequency in a population, and you should be able to explain the biological consequences of, in this case, generally speaking, reduced genetic diversity, but either reduced or increased genetic diversity. What are the possible risks and benefits of those to a population? If you can't do that, you need to maybe go back and rewatch this again, read your chapter, answer the questions in the back of the chapter in your textbook, and absolutely ask me questions if I can help you. Otherwise, thank you very much for watching this, and I hope it was helpful.